The overall goal of this experiment is to analyze assembly of a multi-protein complex, such as the proteasome, using recombinant subunits and non-denaturing polyacrylamide gel electrophoresis, or PAGE.
This method can help answer key questions in the proteasome field, such as what intermediate species are encountered along the assembly pathway of this protein complex.
The main advantage of this technique is that it allows rapid screening of assembly by two parallel approaches, allowing detection of on and off pathway intermediates.
This procedure begins with bacterial expression as described in the text protocol. To perform bacterial lysis, thaw the induced cell pellet, co-expressing the desired combination of proteasome subunits, on ice for 5 minutes. Once thawed, resuspend the pellet in 600 microliters of lysis buffer. Incubate the suspension at 30 degrees Celsius with shaking for 30 minutes to generate the total crude lysate. Next, centrifuge the total crude lysate at 10,000 times G for 10 minutes. Collect the supernatant in a fresh 1.5 milliliter microcentrifuge tube. This is the co-expression soluble lysate that will be used for purification. Similarly, carry out lysis on a second set of cell pellets from bacteria expressing the desired proteasome subunits individually. Mix 600 microliters of total crude lysate from bacteria expressing the desired alpha subunit with 600 microliters of total crude lysate from bacteria expressing the desired beta subunit.